Okay, moving on. So let's just wrap up here with what we were looking at with an individual villus. So we were looking at the individual cells. If you look over next to, and I'm trying to get my pointer going here, if you look over here, you'll see individual goblet cells. Like I said, the goblet cells are those that make mucus. M-U-C. So just think of, you have goblets full of mucus. It's the chief cells that make hydrochloric acid in the stomach, but there are none of those in the small intestine. Now, another thing I want to point out to you is this thing called a lymphoid nodule here. The lymphoid nodules are also called Peyer's patches, and this is a piece of lymphoid tissue where you have white blood cells in a focused area of the small intestine. Why would it be there? Well, to keep the nasties from getting into your body. This is one spot in your body where lymphoid tissue is trying to trap any microbes that made it through the stomach. Trap them, kill them, and make sure that you don't get sick. So the histology, or the slides of the small intestine, again, you have a mucosa. It's arranged with villi, which we already saw. Non-ciliated simple columnar epithelium. Remember, ET is epithelium. You have connective tissue and a muscularis mucosa. So in other words, you have more smooth muscle here. Submucosa, muscularis, serosa. The intestinal glands include our goblet cells, which make mucus, our Brunner's glands, which make alkaline mucus. So one of the things that happens in especially that initial portion of the duodenum is that you have this really acidic compound, the chyme, coming out of the stomach. It's very acidic. So the alkaline mucus then shifts the pH to a more neutral pH, so that by the time you're in the small intestine, the pH is a lot more neutral. And in fact, eventually it becomes slightly alkaline. The intestinal crypts also make enzymes. This is where lactase and sucrase, other enzymes are made. There are hormones that are involved in digestion. These are also the hormones, the satiety hormones are made here. And then Peyer's patches that I was just mentioning in the last slide. I did talk about the villus. The individual villus is really small. There are about 4 to 5 million villi along an individual length of small intestine. The surface area is pretty wide, 200 meters squared, which means you have a lot of surface area for absorption. And most of this is due to individual villi, so each one of these little zigzags, and then all these little zigzags are found on an individual plica. So if you had a plica, each plica would have individual villi. And remember, each of the villi has a bunch of individual cells here through which absorption happens. Each villus has an arteriole, a venule, capillaries within them, so you can see that right here in the vasculature, and also a lacteal. A lacteal is a blind-ended lymphatic vessel. So here's the actual lymphatic vessel here, taking fluids out in that direction. Right here is where absorption occurs. So one of the interesting things is that we have fats absorbed into the lymphatic vessels at the point of the small intestine. So the nutrients absorbed are going to be proteins that have been digested into amino acids. They go into the bloodstream. Carbohydrates get broken down into monosaccharides like glucose, absorbed into the bloodstream. Lipids get broken into glycerol and fatty acid. Those go into the lacteals and then eventually into the lymphatic system and dumped into the heart in the circulatory system. Now on to the large intestine. This is a lot shorter than the small intestine. It's only about 5 feet long. The whole purpose is to reabsorb water and electrolytes here and to compact all of that remaining chyme and undigestible fiber into feces, into waste products. We also do have some absorption of certain vitamins by bacteria, and when that absorption happens, the bacteria actually make other compounds that are beneficial to us. So in the large intestine, in the large bowel, or in the colon, you can also call it the colon, these bacteria are our own personal probiotics. And they really like to eat things that are going to be, that are basically leftover bits of fiber from the chyme that we've consumed. So this is where some of that non-digestible fiber gets consumed by bacteria, and the bacteria produce other compounds that are very healthy for us. The large intestine also stores the feces or fecal matter until defecation happens. There are no villi in the large intestine. Instead, there are haustra, and these haustra are little pouches. They're kind of bumps if you look at the large intestine, and it allows for distension, elongation, helps move the material around for water absorption. In addition to that, there are these large crypts, and they are kind of similar to the intestinal crypts that you find in the small intestine, but they go much deeper into the submucosa, and there are lots and lots more goblet cells here. Why would you need more goblet cells in the large intestine? Well, because you're removing water from the chyme to produce feces. And so you absorb the water in the large intestine, the feces gets kind of dried out, so it helps to have that mucus to move the fecal matter into the terminal end of the large intestine. The large intestine has three regions: the cecum, the colon, and the rectum. The cecum is a pouch, and it receives contents from the end of the small intestine, the terminal end, called the ileum. So the ileum dumps into the cecum, and this is where the variform appendix is attached. And I'll show you a picture of that in a minute. The appendix itself is not functional in humans, although it is an extension that's functional in other animals, ruminant animals like rabbits and stuff. The colon is the large intestine. That's the part of the large intestine, and that's the ascending, transverse, and descending and sigmoid colon. And then you have the rectum, which is the last 6 inches. So in terms of the colon, this is where you have the majority of water absorption happening. You have the hepatic flexure or the right colonic flexure. This is the turn between the ascending and the transverse. So it's going up, that's ascending. Then you have the transverse, and then you have down and out. The splenic flexure is the left or the colonic flexure, and this is the turn between the transverse and the descending. So here's the hepatic flexure, and this is the splenic right here in my little weird drawing. The rectum just temporarily stores feces, and it connects to the anal rectal canal, which is the final portion of the rectum that empties out into the anus. So here we are. Here's the ileum. The ileum has the ileocecal valve here that allows the basically it allows fluid, chyme, what's left over from what we've digested, to enter the initial portion of the large intestine. So this portion is the cecum. Right here is the variform appendix, and of course when the appendix becomes infected and inflamed, if stuff gets down in there, then you end up with appendicitis, and that's really dangerous because if the appendicitis, if this region ends up expanding and bursts, now you have infection draining into the abdominal cavity, and that's very dangerous. If, on the other hand, things go the way they're supposed to, the leftover chyme is going to start drifting upwards through the ascending colon. Each one of these little bumps here are the haustra, okay? The chyme is then transitioned over along the right colonic flexure. We have the transverse colon here. Here is the splenic flexure coming down into the descending colon. Here's the sigmoid colon, and then down into the rectum. In terms of the vasculature, there's a tremendous amount of vasculature feeding the large intestine. This is how water is getting back into the bloodstream, through this vasculature. Over here, down here in C, you're seeing a picture of what the rectum looks like. The rectum has these little things called the anal columns. The anal columns are kind of like a valve trapping any content in this region. And it's not until the internal and the external anal sphincter open that the compounds can leave through the anus. So the histology of the large intestine, there's definitely a mucosa, no villi though. Lots of columnar epithelial cells, lots of connective tissue, muscularis mucosa, which makes sense, you got to push things out. The submucosa, the muscularis, we have circular fibers and longitudinal fibers. Basically, they're going to be arranged in what they call 3 ribbons here. 3 ribbons called the tanae coli. And these 3 ribbons are organized to allow smooth contraction and elimination. The serosa is present here. And then the sphincters that we have to pay attention to in the large intestine are the ileocecal valve. So that's the junction between the ileum, the end of the small intestine, and the cecum, the beginning of the large intestine. And then there's the internal and external anal sphincters. We have voluntary control over the external, but internal is involuntary. And you'll learn more about that in physiology. Now, in terms of other accessory organs, the salivary glands, liver, gallbladder, and pancreas. So basically, salivary glands are going to make saliva and synthesize amylase and lipase. There are 3 pairs of glands. There's the parotid, the sublingual, and the submandibular. And the majority of saliva is 99.4% water. There are some ions, some salts, there's some buffering, and salivary amylase. The liver is the largest organ found in the body. It's enormous. It weighs 3.3 pounds, about, depending on the size of your body. And it is an exocrine organ because it secretes compounds into the bloodstream. We have some work going on in the backyard here. There's a right lobe, the caudate lobe, which is marked by the IVC. I'll show you that in a minute. The quadrate lobe, the left lobe, and then you have the falciform ligament holding things in place. Let's go look at the lobes here in a second. Oh, I guess I don't have a picture of them. My apologies. In terms of liver histology, the lobes are broken down into lobules, and there are about 100,000 lobules, again, depending on the size of the liver, and they're all separated by connective tissues. The connective tissues develop into these little regions called sinusoids. And basically the sinusoids lie between plates that empty into the central vein. So I do have a picture of that. Hepatocytes are arranged inside of these lobules around the vein, which I'll go show you in just a minute here. So right here is an individual hepatocyte, okay? So this region, here's the lobular organization. So this would be a lobule, okay? And then within the lobule, you have the bile ductlets. So this is where the bile is going to be traveling. We have the portal area. You have arteries and veins. We have the branch of the hepatic portal vein. Remember that from the vasculature. Here's a bile duct. And then here's what we call an interlobular septum. The septum is made up of connective tissue separating each lobule from one another. Now, within each lobule, we have a central vein. We have these things called Kupffer cells, which we'll get to maybe in a few minutes. Hepatocytes, which are making compounds. The sinusoids, which the hepatocytes are draining compounds into, which take the compounds to other places. Here's the bile caniculi. The bile caniculi are also associated with hepatocytes that are making bile. We have, of course, the branch of the hepatic artery, a bile duct, which collects bile from these smaller bile caniculi, and then the branch of the hepatic portal vein. So one of the things about the sinusoid walls is that they're fenestrated. In other words, they have gaps, and this allows molecules to enter the area around the hepatocytes. So why is this important? Well, that allows things like the Kupffer cells, which are phagocytic cells, to come into contact with pathogens, debris, or damaged red blood cells, in this case usually red blood cells, to clear them from circulation. So your liver is doing a tremendous amount of work getting rid of pathogens every day and cellular debris. They also get rid of heavy metals that you might have consumed, like tin or mercury. However, having said that, once you get mercury in your body, it's hard to get it out. And all of this would be absorbed by the digestive tract. The liver's function is to make bile. Bile emulsifies lipids in your stomach. You'll talk about that in physiology. It builds and stores down glycogen. Lots of glycogen is stored in your liver. And it's stored there when blood glucose is high. And the liver breaks down glycogen into glucose when blood glucose levels are low. It stores A, D, E, and K vitamins. It inactivates toxins, produces urea, and does lots of other things. All right, it looks like we're going to probably have to do just one more video, so I'm going to stop here.